Great. So what we're going to go through today, I'm going to do this introduction where even if you didn't review Dr. Lewis's webinar already, in Classic, we're going to go through about a 4-minute quick overview of what I think are the key portions of this. And what we're really trying to do is set the framework here. Our focus is on neurological trials, but many aspects of clinical trial design just cut across every type of trial you might be designing. We're also going to have a brief discussion by me where I'm just going to talk a little bit about the framework for designing trials that involve COVID, and also consider how one does research in a pandemic. Think a little bit about COVID trials and studies, and if you're doing existing research outside of COVID, what you might need to do, and think a bit about how you might design future trials now that we've been in a sort of unprecedented situation. After that, we're going to open things up to questions from the broader audience. If the audience doesn't have questions, I came up with a couple pages of things that I will ask everybody to weigh in on and think about, and I might call on people and we will unmute you. So first, I'm going to play for you all a very fast version of Dr. Lewis, one of our other course directors, very excellent webinar from 2017 on asking a very good early phase clinical question. I'd hope to cut this up a little bit more, have this be like the Daily Show where there's just like 30 seconds here, 30 seconds there, but actually there's a good 4-minute clip. So we will work to have even shorter clips next time. I did manage to speed up his speech by 25% without increasing the pitch of his voice, which I was very proud of. So here we go.
So what is an early phase trial? In order to understand what an early phase trial is and is not, it's important to realize that for us to demonstrate ultimately that a treatment is adequately effective and safe so that it warrants clinical adoption, we must be able to define the right population in which that treatment ought to be administered, the target disease or diseases, and those diseases may be characterized both by the general diagnosis or by severity or stage. The treatment strategy, how do we actually apply that therapy? What is the timing, the dose of the medication, the route by which it should be administered? We need to understand the outcome that we are trying to affect. For some diseases, the outcome may or may not be of interest to particular patients, and that may influence whether or not the treatment is warranted. And we need to be able to define a confirmatory trial design that will evaluate the use of that treatment in that population for that disease using that treatment strategy in order to demonstrate a positive effect on the clinically relevant outcome of interest. Early phase clinical trials are designed to reduce uncertainty in each of these areas, the population, the disease, the treatment strategy, and even the outcome, so that we are able to run a successful confirmatory trial. A successful confirmatory trial is not necessarily one that gives you a positive result. It is one that tells you unambiguously whether the treatment is of clinical value in the target population. So we often think about the phases in the development of a drug, but there's an associated series of steps in the development of a device as well. There's an early stage, phase 1 in drugs, and sometimes this is bench work in devices, where we're trying to learn very basic things about the treatment that we are investigating. Then we analyze the results of that work, we make a number of decisions, and we enter phase 2. In phase 2, we leverage what's been learned in the earlier work. For example, the maximum tolerated dose, or the side effect profile, or the pharmacokinetics. And we start to look at outcome results. In after bench work for a device, we may do some early clinical testing to make sure the device can actually function when implanted as it is intended. We take the results of those early stage work, and we make a number of decisions regarding how to design a confirmatory trial, usually called a phase 3 trial in the setting of drugs. But in a device trial, it's often called a pivotal trial, to verify the preliminary evidence of efficacy we've obtained earlier and just demonstrate unequivocally the effectiveness of the treatment to improve the outcome. But this leads to sort of a general concept, there are some stages in development, which we call the learn stage. And then there are some later stages in development where we hope to confirm or verify what we already believe to be true, or at least hope to be true. In the drug development, the learn phase is considered phase 1 and phase 2. And at that stage, we are really trying to answer a variety of questions And so there must be greater flexibility in the number of questions that we ask and the goals. We may be interested in toxicity, feasibility of administration, side effect profiles, variations in pharmacokinetics across populations, etc. Because we are addressing multiple questions and learning in a more flexible way during the learn phase of investigations, we have to have a willingness to tolerate higher error rates, and there may be some sources of bias. For example, we may have a highly selected patient population, for example, one that has failed prior treatments, and that may lead to bias in the estimate of a treatment effect or side effect profile relative to the effect size or the side effect profile we would see in an unselected population. But it is important to understand that phase 1 or especially phase 2 trials are not just small, underpowered confirmatory trials. They actually have different goals. In contrast, during the confirm phase of investigation, so this would be termed a pivotal trial in device work or a phase 3 trial in drug work, we want to have a rigid control of error rate. So we'll have a very rigid control of our type 1 or false positive error rate, a defined power, meaning the probability of getting a positive result if the treatment has a specific and pre-specified treatment benefit. We're going to pre-specify and be held to that pre-specification in defining a single primary question or outcome that defines whether that phase 3 trial is positive or negative. So this is the setting in which we often use a traditional one-to-one randomized control.
All right, and that takes us through the sort of beginning intro. I think a tour de force there. I think to really sort of think about that is when you're doing an exploratory phase trial, you really want to answer a question that gives you a quantitative guide to what's the next step you take. And just for those of you who may have joined a bit late, if you have a question, if you post it in the chat room, I will work to try to address them, but then we will be opening things up to the broader audience. I know some of you have called in on the telephone only. We don't have a great way to unmute you. If you do Twitter, you can, I guess, direct message me at Will Meurer, WILLMEURER, and I'll try to answer the questions that way. So this is a really good way to design trials. This is how we start out the clinical trials course each year, and the world has changed. So thinking about this, if we were, let's take a step back and say right now we're working to do new research on COVID. I mean, particularly through the lens of neurologic trials, back to the basics. When you're designing research, clinical research on humans, whether it's through NIH or NINDS or other institutes, you really have to think about looking at preclinical evidence first. And in a new disease, where does preclinical evidence come from? In the case of COVID, there's some preclinical data on things like SARS Classic or the original SARS or other coronaviruses, MERS. But there's also preclinical data on similar disease pathways, such as ARDS or cytokine release syndrome. So looking at what we understand about the biology and the pathophysiology can help inform how we design trials, because we want to make sure we're designing our studies based on actual science and reasonable biologically plausible hypotheses. The other issues are how one can do research in the pandemic. Right now, there are a number of things that we need to do. First, we need to take care of the patients out there with this terrible disease. But second, we do have to protect our staff and our research staff, and balancing the potential for benefit in times from research, the potential for benefit for future people to learn how to take care of future patients can be tricky to all sort of layer up and figure out when certain types of studies can start and what certain types of studies can keep going. And it's always important to learn, but the types of learning that we can do that help treatment can often be more, prioritized. That's sort of couched with the standard issues that oftentimes when we start a clinical trial, we have a lot of unknowns. And when you have an unknown disease, you have even more unknowns. And it can be challenging to design the smaller studies to lead to the later studies when there is such a huge public health push to discover cures to something that is causing a great deal of pain worldwide. in terms of designing COVID trials or studies, again, thinking to similar diseases, but trying to learn as much about epidemiology as you can when developing these things. I think definitely there are emerging reports in the neurologic space, and thinking about, is this COVID causing new neurologic symptoms, or is it COVID causing problems with people who have chronic or existing neurologic disease, and how those studies might be differently designed. I think if you have existing trials that are ongoing, particularly in the exploratory phase, you have to think about whether your protocol can be adjusted to continue to answer your primary question. Many early phase studies have a lot of extra data collection to learn about other proof of concept aims and other things like that, some of which might not be able to be done given the balance between protecting staff and avoiding contact with patients. But some specialized neurologic assessments may be able to be done in certain situations. For example, in stroke, the NIH stroke scale is often administered via video for people who are doing telemedicine for clinical purposes. So how it would perform on video is well characterized, but many other neurologic assessments have not been performed in those, and would be very challenging to do without you being in the room with somebody. Thinking about how you develop the trials of the future, we should not stop doing clinical research. So we have to focus on doing good science, asking answerable questions, but answering the right question for where the science is at that moment. We want to come up with designs, though, for the future that maybe are robust to the circumstances. If we are, for example, I'm leading a text messaging study for hypertension, we are able to get outcomes from people because we give them blood pressure cuffs to use at home as part of the study, and those can be mailed so that it doesn't require direct patient contact. So we were hoping to have in-person visits to better calibrate the blood pressure cuffs and get more accurate measurements. But in this time, we can still answer our fundamental question of, does our text messaging intervention reduce blood pressure with, sort of circumstances that have changed somewhat compared to how we started the trial. And you do potentially have to make statistical adjustments if there are unforeseen circumstances like a pandemic. But you want to design your future trials perhaps with ways that they could be done if people cannot have much, if any, in-person contact. I think this has really illustrated that this is going to be with us for a while, and things like this can pop up again. But we also want to think really critically about everything we collect. If everything we collect is potentially putting somebody at risk, do we need to do all these blood draws and so forth just to sort of find things? There's good reasons biologically why blood draws are needed. That's great, but I think we have to be more cautious about avoiding research waste. So that was the set amount of stuff that I wanted to just sort of cover as the quick overview for this session. As I was hoping, I tried to get through a lot of ground in the first 15 minutes. I do have a bunch of additional areas for question. But first, I think I would possibly call upon, and we can certainly unmute you, or you should be able to unmute yourselves if you're on the app. Any of the other CTMC course faculty or directors who are on, who have anything they want to add to that overview. I think we can unmute Lori.
I can unmute myself. I'm not sure I have a lot to add. Did I unmute myself?
You did.
Okay. I guess one of the things that is of interest when you're doing these studies, can you hear me okay? Sorry.
Yep.
I think one of the things that's of interest when you're thinking about these studies too is the whole idea of trying to do things by remote monitoring, and are they validated? I mean, you mentioned that on the NIH stroke scale, it's definitely validated, but other studies, as we look at them, how do you go about validating something while you're doing a study? Because you don't really necessarily have time right now, right, to go through a whole validation. So I'd be interested in your thoughts on that, Will. But I think that's one of the issues. So it looks like there's a question too. Were you going to say something?
Chris?
Yeah,
go
I was
for
going
it,
to
Chris.
say, I don't think that's possible in an interventional study to validate any type of measure because you're changing the natural course of how that would do, and you can't disentangle treatment effects from validation progression on the endpoint that you're studying. So I think one of the challenges is that sometimes people try to do that and it causes issues, and you really need to disentangle those two things.
Right. Yeah. Yeah.
I think just to quickly address the question about the precedent for virtual informed consent. Yeah, actually REDCap has a module. There are other modules. Some places use Sign Now for you to do telephone consent and get a electronic signature. We've been using that in the ongoing trials in the Siren network. We have it. Our standard process for getting signatures is on an iPad that's actually taking a link into REDCap. But that REDCap link could be sent to somebody who isn't actually physically present, and you could either use video conferencing or telephone to explain the study to somebody who's, we call this remote e-consent. And, and Dr. Coffey, who just spoke, brought up when we were talking before, there's a really excellent FDA guidance on, how to adjust research during the pandemic. And it goes actually into the weeds on this to the point where you could throw, like if it's even if it's a room and you can't go in the room, you can slip the consent under the door, have them take a picture of it and send it to you. Or if you email that to them, they can send you a picture of the signed page. There's a lot of flexibility that's allowable on that so that consent can occur without in-person contact. And Dr. Conwood also points out that some outcome measures are already validated for phone. The modified Rankin, she has the ALS-FRS as another one. But I think in terms of doing new validation, if it's your primary outcome, I think that as Chris points out, there's no real way to do that. But it does point to us for some other types of. There are certain types of tests, like, for example, secondary outcomes in a clinical trial of cardiac arrest that I'm going to be, that we're working to launch right now, that involve, very detailed cognitive testing. And there aren't valid ways to do that without people being in person, at least for the measures that we've selected. But it's an area of, and for certain things that test reaction time and so forth, without knowing what sort of latency there is in terms of people's internet connections and things like that, those things can be very, very challenging to get actually good characteristics on. And it's funny that I have the sort of the random people whose faces are showing up for me the way that they are organized currently. I see Dr. Levitt from New York as a neuropsychologist who had been in the course, and I'm sure she understands that, sort of what we're going through, like even in terms of very classic neurocognitive tests like trails, how you do that, how you could even think to do that over FaceTime or things like that. It's not currently something that's been tried, and it's not, and when it's been tried, it hasn't gone all that well. So it really, oh yeah, go ahead, Lori.
One thing I just wanted to say about e-consent with REDCap is that for some trials, you have to have REDCap part, you have to have it part 11, what I'm
Ready,
blocking on.
compliant,
21 CFR Part 11.
Yeah, so you have to have for FDA trials, if you're going to go for FDA approval. So some trials won't be able to switch over to e-consent for everybody if they don't have that form of REDCap. But the
standard REDCap doesn't comply.
Right. The standard REDCap doesn't comply.
But the most sites are set up in a way that it can be built in such a way that it is in compliance, which is, it's a matter of which buttons are clicked by your institution in your REDCap instance. So it is potentially compatible, not compatible, at default, depending on the setting. So we have set those settings correct. We have.
Will, can I say something about validation first to make a point
Oh
before
yeah,
you get
for
off
sure.
that topic? One thing that I think is helpful just to remind people of, because I think this often gets lost in discussions, is even if you're using an instrument that has been validated, if you change anything about it from the way it was validated, it's no longer validated. So it's the whole process, not just the score that you're getting from it. It's the whole process and how you give the instrument that is part of the validation. And I think that's a subtlety that gets missed if you, like, some questions don't apply to your specific area, so you leave certain questions off of an instrument, it then no longer is technically a validated instrument.
Or even if you have to be face to face, right? I mean, to
Translation, if you translate it to another language, you have to revalidate it. I mean, I think all these things often get overlooked in many cases when somebody's trying to like quickly implement it.
I think there's a good question from Dr. Ballou about studies that are evaluating, I guess, novel monitoring strategies, and if that monitor, what's the cleaning procedure for that monitor? Can it go into different patient rooms, or is it a monitor like, let's say, like a pupillometer or something, where you're going to actually take it, get some readings on one patient, and take it over to the next patient? In many cases, with there being people who are either COVID positive or not, we have single-use stethoscopes and things like that. So could you move monitoring devices? I think that's an interesting question. Right now, at least at my hospital at the University of Michigan, they're screening everybody who's in the hospital regardless of their symptoms, which means that at least in terms of within the accuracy of the nasal swab, you should know everybody who should be shedding virus, at least on the patient side. I guess the staff are another matter, but that will probably come as well at some point. So I think, that type of research is on hold. Currently, most research at Michigan is on hold if it involves direct patient contact, but isn't an interventional trial that offers the prospect of direct patient benefit. And I think some device-based studies could potentially fall into that category, but right now, at least that's how our institution is prioritizing the sort of return to the sort of research normal. But I think that's a good question. Certainly, there's certain procedures, for example, if your study requires LP, that it may be very difficult to do research LPs currently. If you need to do research imaging in terms of, patient comfort tests, cleaning an MRI scanner, if the person's viral status is unknown, a lot of open, questions. And you could say, well, we have everybody wearing masks while they're in the hospital, but I guess I wouldn't want to sit through an hour-long research MRI wearing a mask the whole time if I was a little claustrophobic to begin with. I think that would be really hard. So I think some of those issues are things to think about from a strategic and procedural perspective. I think with Dr. Ballou's question, I think there's also another sort of, even though that, he's talking about monitoring strategies for devices, I think when one thinks about certain clinical trials, perhaps you're performing them as a site investigator, whether typically you would have monitors from the clinical coordinating center, the contract research organization, coming to do source document verification and monitoring your clinical study. And a lot of those activities are currently on hold or being moved to remote monitoring, where outside folks have access to the medical record and can help do that. In our Siren network, we offer that. Many institutions, some institutions are very open to it. Some it's a little more of a uphill question, but I think right now all institutions that are doing research are probably being quite open to remote document verification in some form. And obviously that can come in a variety of ways, whether it's PDFing somebody's entire medical record and putting it into a secure file location for monitors to look at, versus giving sort of very limited view-only access to a health record through a virtual private network. It depends. So I guess I would be interested in if anybody out there has a institutional triage process determining what research studies are currently on hold and when other research studies may be reactivated, because I think that's something that's very, very important to a lot of us right now. And I think it's something I'm on the committee that's working to set up that strategy for U of M. But I would be interested to know what others are doing in that space in terms of figuring out when research can be turned back on and what research and how that's determined. Lori, how do you do it at Iowa?
I was just typing into the chat box, so I guess I could talk instead. But so we actually set up a departmental oversight committee that would to decide these things. The Office of Research for the university has a university-wide no research, no ongoing research, but has a caveat for clinical research that's felt to be critical. And you can get approval from them. Sorry, my phone is ringing for some reason.
They're approving another one of your studies.
Yeah, you can get approval from them for depending on the situation. And we have actually started recruitment for another study that is currently a device study that is doing a comparison, head-to-head comparison of intervention for stroke. And that one we did open up because we could do remote consenting. We could do, it didn't entail a research assistant coming in. All the data could be put in remotely, and it didn't entail any other personnel in general or change the flow. It just meant using one instrument over another. So it's just a comparison, device comparison trial. So we did reopen that one, and immediately had 3 people who needed thrombectomy who came in the next day, which was just a fluke. 2 of whom got recruited into the study as new patients. So everything in that study can be done remotely. So the oversight committee includes our 2 directors, the coordinators who are head of this, because I think the coordinators have a lot to say about what the risks are to the personnel, which I think is part of this that we have to keep in mind, and I think you mentioned it. But it's an important part to keep in mind. And then myself, the head of the stroke program, and our head of associate dean of research for the College of Medicine, who also happens to be in our department. So the 5 of us meet every week and sort of review where things are. We've had discussion about, there's a natural history study for people who have torticollis who are coming in for Botox. And do you open that study up? Because the patients are still coming in for Botox, right? They're already coming in. Well, the debate on that one is that requires a clinical research nurse to come in and administer testing. It requires the patient to stay longer than they would normally in the institution, although they're already there, and it may increase their exposure to other people than the few people they would be exposed to if they just came in for clinic. So I think there's a lot of factors that have to be considered when you're reopening things, and we're doing it in the department sort of on a case-by-case basis. But we have to, before we reopen something, also we have to get approval from our IRB, because our IRB has, actually they've stamped some of the, and I think Hina is on this. She's one of the people on the committee with me, but they stamped some of our IRB consents that they're on hold for right now. So the IRB has got to be involved in this decision too. If I misspoke, Hina, you can certainly type in the chat box.
It looks like Dr. Orta from Henry Ford was talking about they're having some clinical trials in the in the neuro-oncology space that may be opening up for people who do not have any other therapeutic option. I think that's a sort of a great question too. I think it goes to what Lori was talking about. In certain cases, cancer chemotherapeutic trials regimens may have very standard care, and a new regimen may have very overlapping blood draw requirements afterwards and so forth. And the amount of what it looks like is different to be what's different about being on that versus a different like standard care versus the investigational drug. I think some of it may have to do with when there's complications, getting information from the patient and the other staff, but a lot of that could potentially be done remotely. So I think that's really something that you have to think about. And I think also, if you are if you were doing a torticollis study, if there are a lot of like patient-reported measures or things like that and parts of the scales that need to be collected, how much of that could be done remotely to limit their time in there? If it is a scale that's sort of valid for self-administration, might not be.
I think the other thing is what things can you is the sponsor willing to allow you, or are you willing to drop to have missing data for certain and these are for like return visits. Are you willing to have missing data for things? I think Erica brings up an important point, though, the whole issue of when there's no other therapeutic option. There's been the discussion about for ALS trials, for example, if an ALS trial comes along, would you open that up because it's a short and important time frame for the patients, and so we've had a lot of discussion about this. We don't have an ALS trial that's ready to go at this point, but depending on how long this shutdown continues, we may be faced with that question too.
Dr. Coffey, did you want to talk about what sorts of statistical considerations one might have to take if you, for example, had to change or modify your primary outcome measure?
Chris, you're on mute.
Possibly he maybe had to leave. I don't know if that's when he's teaching that class of the undergraduates. I think that might have been. So he's probably teaching undergraduates, or not graduate students, how to answer that question right now. I think the key is likely to be consult with your statistician early, be transparent, make these changes in public. And if your trial should be registered on clinicaltrials.gov. I think if any of us have been out there kind of looking through clinicaltrials.gov on maybe some of these new trials that have been coming out, one thing that I think we can all do is try to make sure that what we have in clinicaltrials.gov about our studies is understandable, so that somebody could figure out what kind of analysis we were planning to do, and if there are adjustments to it, how those adjustments were justified and made. If you're doing COVID-related research, there's some journals that will actually take protocol papers right now that typically have a publication fee, but are waiving that in the case of the pandemic. But even if you're not doing a COVID trial, posting updates, if you have written a protocol paper that describes your protocol in detail, posting updates to that as soon as possible. I work on an editor on a couple of clinical trials journals, so I've been seeing some updates about how these plans are changed. So being transparent and kind of doing these changes, I think the important thing generally is to make those changes based on the conditions, not necessarily the in response to your data. You're making the changes to your primary outcome in response to the external conditions, not how things are looking with the data. If you change your primary outcome because you've looked at the data, that's usually problematic in terms of how you can interpret things. So it really, it really is an interesting time to be doing clinical trials. It's tremendously important, but as with many things right now, there are unique challenges. Another challenge is just there's a rapidly changing standard of care. I think, for example, for cardiac arrest, because in out-of-hospital cardiac arrest, patients, it's unknown what their COVID status is, so it takes longer before definitive care can be delivered to patients, like with a defibrillator or paramedics administering drugs because of the timing that it requires to get safely ensconced in personal protective equipment. So that's changing what standard care cardiac arrest treatment looks like across the country. And then, if you're studying a disease like COVID, the standard of care could be changing every couple weeks as new treatments come online. So if you have a control group there, it can be, you have to think about flexibly in advance how your trial is going to adjust or handle those things statistically. So there are, a lot of things to consider when you're either adjusting your current trial or planning your trial of the future. And again, with our focus in the clinical trials methodology course on designing neurologic trials, I think there's also, it's key to focus on learning about the diseases that we've been studying, but doing so in an environment where we are kind of constrained in what we can do, but we want to think as scientists and be as, upfront and thoughtful about what we're doing as we go forward.
I think, another question that I would be interested to hear you discuss would be how the bias of the patients that we recruit into trials during this time period as well. So, for example, the acute stroke trials, we know that patients maybe are waiting until their symptoms are more severe, or so you may be or people who are willing to come in to be in a trial may be a different group than you would normally be able to recruit into a study, including those who can participate in remote trials, right? Because at least in our state, not everybody has the same kind of internet access, and their ability to do things online may be extremely different. So I don't know what your thoughts are on that. So there.
It's a huge issue in terms of equity, right? And if there's this differential access, if things require video visits, I mean, this is a subset of many of us have children in schools. Broadband access to sophisticated video devices is not evenly distributed across the country or the world. So if we do studies in the future, but we need to do a video visit, and we're only selecting people who have faster data plans and faster phones, we are selecting for a population that is more privileged. And as such, we do know that many diseases affect the less privileged disproportionately badly. So a lot of opportunities for bias. And I think we when we were redesigning aspects of our data collection for our text messaging hypertension trial, we struggled with this even before the pandemic because we were trying to improve our retention, but we wanted to give the people a lot more opportunities. In this study, we enroll people in the emergency department. We did so in person before, although but not yet could do it remotely after the patients have gone home. And we would hand them blood pressure cuffs and text them for a few weeks to see if they were still hypertensive. Then if they were, they would get randomized to a series of behavioral interventions by text or interaction with our research team. And as we looked at, we were going to have 6 and 12-month visits in person to collect more, blood pressures on more expensive blood pressure devices that are considered more accurate than the blood pressure cuffs we were giving them for their home use. But when you design a study like that, sometimes people are like, hey, why don't you give everybody Bluetooth enabled blood pressure cuffs? Because then things will get collected directly. But then the people in your study, and our study was focused on a less privileged group of patients in Flint, Michigan, then we're selecting for people who have better devices. So we went with just text messaging, which is a sort of more egalitarian type technology, so that people could easily respond to us. But as we went to remote visits, we had been thinking about, well, should we try to get people on video so we can watch them blowing up their blood pressure cuff so that we knew they really did it for the incentive? But not everybody can do that. So we've accepted a variety of ways, like taking a picture of it on your wrist and so forth, that we've been more flexible about that. So I think those things have to be sort of considerations, but I think it is extremely important to consider sort of the equity in these things, and that things that you can collect from the largest group of the population. So
yeah, I think the equity and inclusion issues are huge, potentially huge for some studies. The next question is a good one, and I was just going to bring it up as well. So the next one talks about quality of life outcomes, or outcomes that could be affected by the pandemic. We have another trial that looks at sleep and the use of a sleep aid to help with mild cognitive impairment. And people's sleep is definitely changed. I don't know if I have one more weird dream. I don't know what I'm gonna do. The other night I dreamed of somebody poisoned me. And I woke up in the ICU. I mean, so sleep is definitely disrupted already. And so how that might affect what's happening. The question of quality of life measures. And I don't know if Victoria has something to say too, because she is all involved.
Well, first of all, call me.
Yeah. Me? Because I'm being poisoned? Help!
No, for psychological help. Yeah, no, I mean, I think it's a really interesting time. All the things that I'm most interested in are now sort of coming to the fore for everyone. The quality of life measures and ways of incorporating those into what we're doing, as well as sort of patient-oriented outcomes. I know that PCORI has been putting out some new RFAs, but I don't know. I'm not sure, Lori, if you had a specific question for me.
how much it's probably more along the lines of attribution, right? How much, if you're doing quality of life measures, how much can you attribute to the disease in question and tracks on it, and versus sort of what would be a norm, like for maybe a neuroqual or something that has population-based norms based on age. How much of a hit on it is coming from their disease, and how much of it is coming from people who are relatively non-diseased, but just I actually, as I've been working, this cardiac arrest trial that I'm the PI of, we're working on sort of implementing the training for our patient-reported outcome battery using NeuroQual, and I keep like doing the questions myself, but I'm trying to answer them honestly, and I'm under the population expected mean, because COVID's affecting my quality of life. So
another thought would be, how do you look at the population before, if it's a long-standing study that you've been recruiting people over a long period of time, or you are following people longitudinally? How do you compare the data before COVID and now? And I don't have any answer to that. I'm full of questions today.
That's a super relevant question for me. I'm actually in the midst of my follow-up, my 3-year follow-up from a cohort study that I've been running, and we've had to discontinue. So now a lot of those measures, I mean, yeah, what are we going to make of them? How do we adjust for COVID-19?
I don't know. Would you have to do a sub-analysis? I mean, I don't know.
yeah, I mean, I think they're probably designing the statistical analysis plan for this this may not be the form that we could do that, but you could certainly consider that there may be people out there, at least for some of the more common, neuro-related patient-reported outcomes, doing some population-based stuff to try to get a sense as to, in people who haven't accumulated new medical problems, where things are tracking. It seems like it's really tough, though, right? There's things that seem like they would be like common sense to be doing, but research is so distracted in general and focused on, keeping society alive that maybe there hasn't been time to answer those questions. Although, with people in a variety of different places, everybody has different bits of bandwidth, but certainly there could be both clever ways to do that with your existing data, but hopefully some calibration cohorts or cooperation across studies to figure out what's the correction factor that needs to. Dr. Levitt again.
Can I jump in, Bill? I mean, at our center,
Yeah.
we're doing something pretty cool. We've initiated a survey-based study to monitor our MS patients. Our MS center is doing this, and we've sent it out via REDCap. We have 1,000 respondents, about half of whom have MS, and the other half of whom are some sort of controls, non-MS controls. And it's a lot of PROs, it's a lot of mood measures, but also a lot of other sorts of questions about symptomology and, just so, and this is something that we pulled together across 5 centers. So I think I'm heartened to know that we're doing that. And I know that there's lots of things like that taking place, lots of initiatives that came together really quickly. My sense, anyhow.
We'll need to just apply the Levitt equation to our PRO data. And at the other end, it will magically come out as adjusted for. Yeah, but definitely.
You can also help us all with our stress.
Definitely.
All right. I think, at this point, I've covered all the high-level topics that I wanted to cover. I will certainly open it up. People who have the ability to, I guess if everybody unmutes themselves and asks a question at the same time, I probably won't be able to understand that. But if you want to unmute or type something in the chat box, I think we can try to cover other things. Well, while people are thinking about if there's anything they want to put in the chat box, and again, it's totally fine for us to end a little bit early on this one, just to give you some other things that we're going to be covering as the course goes by, or as the ambulance goes by my house. In the next setting, we are going to be reviewing Dr. Thebane's pilot and feasibility study webinar. We won't be thinking as much, but I think we'll have to continue to think about how the pandemic affects these sorts of studies. These studies are very important. They're very hard to do at baseline because you have to answer hard questions with few patients. Now you have to answer hard feasibility questions with few patients and a rapidly changing environment of how you can even do research. So I think it will be another really important session. The overall goal here is to provide a sort of general curriculum and discussion of the topics that we would often go through in the CTMC, so future discussions on sample size and those things. We're going to work to try to record that sort of quick 15-minute summary at the beginning, and we will record the entire thing, but the sort of high-level overview at the beginning we'll make a recording of if people want to listen to it while they're exercising or doing other things to sort of maintain their own sanity. I go walk down the trail by our house during our faculty meetings, and I have like a selfie stick that my phone goes on basically so that I can be on Zoom as I walk down that. So I really hope everybody continues to focus on moving science forward, but staying healthy and mentally well through this time. Dr. Gutman, anything else you wanted to sort of give as sort of procedural information?
No, I just think, just a reminder that this year the clinical trials methodology course has taken, due to the pandemic, a completely different format. And so we welcome everyone to join these. These are open. People have asked before webinars have always been open, but we're specifically hoping people will be able to take advantage of joining these into the fall. So right now we have a schedule that goes into July. We'll be expanding that over time.
And we're working to really.
Karaoke.
And
So
I mean, what's the plan?
I've been doing some feasibility studies, and we'll have something in place by July.
we might
I
just
use
have
one
to have
method
the
that
alumni for
failed.
karaoke, but because it could be disastrous, but well, it usually is
should be.
disastrous.
It must be.
It always is disastrous. All right. Thank you,
Yeah,
Will.
no.
This was great. Thanks,
Yeah,
Will.
and again, please, everybody, in the chat box, there is a evaluation link for this. Please give us comments. We're gonna revisit things after we've done about 2 or 3 of these to try to pivot this and make this maximally useful. We may do some things in other formats, but we're gonna kind of keep this format here for a bit. But the more you can put into that as to what would make it better, we're going to continually try to adapt them. And again, really, really a pleasure to see everybody today and talk to you all about this. And again, please don't hesitate. And we're really sorry we're not having the course this year together, but we're gonna try to do some interesting things to try to keep your research moving forward. So with that, I think we will end the session, and I have a moment for lunch before my next call. Talk to you.